Approximately 1% of the world population is affected by intellectual disability. The vast majority of ID cases is due to monogenic causes that follow a Mendelian inheritance that is either dominant or recessive. While the prevalence of AD-ID is comparable all over the world, the number of AR-ID mutations is considerably higher in certain regions of the world. These global patterns have been investigated previously, and the epidemiological results revealed that high AR-ID prevalence is especially observed in countries where marriages with members of the same family are particularly common. Such partnerships are also called consanguineous. For example, Martin et al. Recently showed the impact of different mating schemes on AR-ID prevalence within the same healthcare system, the NHS of the UK. Using data from the Deciphering Developmental Disorders study, they found a higher prevalence of AR-IDs in patients with Pakistani ancestry who are characterized by a higher rate of consanguinity than in patients of European ancestry. So, what is the connection between AR-ID prevalence and the mating scheme of a population? Independent of the mating scheme, the prevalence of autosomal recessive ID is determined by the deleterious alleles that enter the system by the de novo mutation rate on the one hand, and those which are removed from the system by natural selection on the other hand. In a steady state, these processes are in balance, and if they are the same for two populations, so should the prevalence. So why does the mating scheme matter? Crucially, populations all around the world are far from being in a steady state. In particular, the number of humans on the planet has exploded over the past centuries. Therefore, we analyzed the impacts of different mating schemes on AR-ID prevalence during and after a phase of population growth, an aspect which has not been sufficiently studied theoretically so far. As the name says, in random mating, the partner is drawn completely randomly. As population size increases, so does the choice of possible mating partners. In a consanguineous population, this is somewhat different. Here we have a couple. For a better representation, we reduce the view slightly. They have 3 children, 2 male and 1 female. The oldest brother and sister will have children in turn. These children are cousins. When the cousins mate, it is called a 4th-degree consanguineous mating. If siblings were to mate, it would be a 2nd-degree consanguinity. With increasing population size, the selection of possible partners remains almost the same in contrast to a random mating population, since the partner is still mainly chosen within the same family. Now a few explanations on terminology of population genetics. First of all, when does an individual fall ill with autosomal recessive ID? The cells of the human body are diploid. That means that there is a double set of chromosomes with a total of 46 chromosomes. 2 copies of each of chromosomes 1 to 22, the so-called autosomes, and depending on the sex, 2 X chromosomes or 1 X and 1 Y chromosome, the gonosomes. There are about 1,000 known genes that can be responsible for autosomal recessive ID that are equally distributed over the chromosomes. Let's take a closer look at one such gene. Mutations can occur at any position in a gene. There are neutral variants and potentially disease-causing variants, shown here in red. An individual will develop ARID if there is at least one deleterious variant on both gene copies. If the variant is located at exactly the same position on both copies, this is called homozygous. If the variant is located at different positions on the 2 gene copies, this is called compound heterozygous. In our simulation, we visualized the number of potentially harmful mutations by using different shades between yellow and red. If an individual has no AID mutation at all, then it is yellow. The more potentially disease-causing mutations are found in their genome, the more the color shifts towards red. If a mutation is homozygous or compound heterozygous, the individual falls ill, which is represented by a black border. In our model, an individual with ARID cannot reproduce further and thus cannot pass on mutations to the next generation. Another important term from population genetics is the mutation load, the mean value of mutations per individual. Since our population will grow, from now on we will only show the heads for a better overview. Speaking of population size, our population of initially 1,000 individuals grows to a final size of 10,000 in about 120 generations, or roughly 3,000 years. These parameters were chosen to mimic a scenario resembling the early growth phase of ancient humans. Simulations of a larger population or migration are computationally impractical. Let's get to the simulation. I will explain the diagram to you step by step. As just mentioned, the population in our simulation grows over 120 generations, from generation 500 to 620. Since we want to find out how many generations after growth a steady state is reached again, we observe a range of 2,000 generations in total. Thus, the phase of population growth in our diagram shrinks a little. The mutation load, which is the average number of mutations per individual, is plotted on the right y-axis. The prevalence, defined as the percentage of individuals with ARID in the overall population, is plotted on the y-axis on the left. We worked with a forward genetic simulation framework SLIM for a wide Fisher model, which contains that the generations do not overlap. In other words, the generations are distinct. We achieved comparable results with an alternative approach based on an adaptive dynamics model and the Gillespie stochastic simulation algorithm. So let's see what happens when we run the simulation. Here on the left, you see the exact parameters during the simulation, as well as a small representation of our population will appear. With the beginning of the growth phase, there is a sharp drop of the prevalence. Interestingly, this temporary dip is accompanied by an increase of the mutation load. The level of the steady state is only reached many generations after the end of expansion. So this was very fast, and it's difficult to keep track of all parameters at once. So let's rewind and start again from the beginning and analyze it step by step. In the first phase up to the 500th generation, the population size remains constant. We begin with a healthy population of 1,000 individuals. No individual has any ARID mutation. Over the first few generations, the mutation load increases due to de novo mutations, and thus the prevalence also rises until a steady state is reached. In other words, per generation, the number of mutations that enter the population due to the de novo mutations is the same as the number of mutations that disappear from the population due to natural selection. Now, let's take a closer look at the phase of population growth that starts in the 500th generation. With the beginning of the population growth, the mutation load starts increasing again, while there is a strong decrease in prevalence. Why does this happen? Let's try to illustrate it with some of our well-known figures, now again including their bodies. In this small population, this individual has an ARID mutation in a gene. Coincidentally, another individual in this population also has a mutation in exactly the same position. Now, when these two mate, their child has a 25% chance of autosomal recessive ID. The same applies if the two individuals have a mutation in the same gene, but at different positions. Remember, the child would be compound heterozygous. If the population starts to grow, the probability that these two individuals will meet decreases as the number of potential mating partners increases. This also reduces the chance of their mutations being removed from the population due to selection. Since this applies to all ARID mutations in the population, the mutation load increases while the prevalence decreases. Now we move on to the third and final phase of our simulation. As the mutation load increases further and further, the prevalence eventually starts to grow again and approaches a level that is similar to the one before the population growth. At the same time, the mutation load also stabilizes, but on a much higher level than before. How can these observations be explained? Let's go back to the illustration. There are various ARID mutations in different genes distributed throughout the population. Due to population growth, it is less likely that any two individuals with a mutation in the same gene would meet and mate. Therefore, the mutation load increases, there are more individuals in darker shades, and the prevalence is low. There are few individuals with a black border. As the mutation load rises and autosomal recessive ID mutations accumulate in the population due to the de novo mutations, more individuals share mutations in the same gene. So the chances of two such individuals mating increases again, and the prevalence begins to rebound. Over hundreds of generations, this process continues, until the prevalence eventually reaches the level prior to population growth again. At this point, a new steady state is reached. De novo mutations and selections are in balance again. However, the mutation load of the population stabilizes on a much higher level. So these are the patterns in a random mating population. But what is different in consanguineous populations, where empirical observations show a higher prevalence of autosomal recessive IDs? The simulation shows no relevant effect of an increasing population size on mutation load and prevalence. Which makes sense, because the choice of possible mating partners does not depend on population size in a consanguineous population. A direct comparison of the two diagrams reveals a huge gap between the mutation loads of the two populations, while the prevalences are the same when the steady state is finally reached. Since the chances of two individuals with the same mutation mating never changed in the consanguineous population, no ARID mutations accumulated. The mutation load remains at a constant lower level. In a sense, the population size doesn't matter in this case. So, what can we learn from these simulations? As mentioned earlier, over the last few centuries, the world population has grown exponentially. But projections indicate that this growth will slow down over the course of the 21st century. So, in terms of our simulation, we are about to reach this point. And the results suggest that the prevalence of ARIDs should therefore be expected to increase in randomly mating populations around the world, even though this process will take centuries. Thank you for watching to the end. We are very happy to receive questions and feedback on this first video. If you would like to see more, then give us a like, subscribe, and get in touch with us via Twitter, ResearchGate, email, and so on. You can find the links below this video. Thank you.